Till today, according to publicly available reports, nobody had given the public a specific technical on-the-record explanation for why. Today, that changed. The woman who has, according to publicly available record, personally helped contribute to solving more than 300 cold cases using this exact technology just went on the record and explained the obstacle with a level of precision that no press conference, no sheriff's statement, and no mainstream coverage has provided. What she said is specific. What she said is verifiable against available forensic science. And what she said changes how this entire investigation needs to be understood. Stay with us. Because the answer, when you understand it fully, is not what anyone expected. And because embedded inside that answer is something else. Something that could potentially change the timeline of this case in ways that most people are not yet aware of. This video is based on publicly available reports and expert discussions surrounding this case. Where analysis moves beyond verified reporting, it will be clearly labeled as such. Every claim presented as verified is attributed to its source. Every theory is labeled a theory. Every single source claim is identified as such. Before we get to exactly what was said publicly and exactly what it could mean for this investigation, you need to understand how investigative genetic genealogy actually works. Because the obstacle that has reportedly been identified only makes sense once you understand what this technology actually requires. Most people who follow this case are familiar, according to available reporting, with CODIS, the FBI's combined DNA index system. When a forensic laboratory processes DNA evidence from a crime scene, they generate what is called an STR profile, a short tandem repeat profile, which maps a specific set of genetic markers. Those markers are uploaded to CODIS. The system searches for matches against a database of profiles from people who have been convicted of crimes or arrested in jurisdictions that require DNA collection. CODIS is, according to available historical records, powerful. It has reportedly been responsible for millions of investigative connections since its creation in the 1990s. But it has one fundamental limitation. If the person responsible for a crime has never been in the criminal justice system, never arrested, never convicted, never had their DNA collected by law enforcement, CODIS has nothing to match against. That person is, according to available analysis, invisible to that system regardless of how strong the physical evidence may be. Investigative genetic genealogy reportedly solved that problem by changing the question entirely. Instead of asking whether the crime scene DNA directly matches any known offender, genetic genealogy asks a different question. Does this DNA match, even partially, anyone related to the person who left it? Because DNA is inherited, because you share portions of your genome with your parents, your siblings, your children, your cousins, your second cousin, even your third and fourth cousins. A database of unrelated people who happen to share common ancestors with a suspect becomes, according to available analysis, a potential map that could lead investigators toward that individual. But here is the critical detail that most coverage has not adequately explained. Investigative genetic genealogy does not use STR profiles. It reportedly requires an entirely different kind of genetic data, what is known as a SNP profile, single nucleotide polymorphism. A SNP profile is generated according to available scientific descriptions by analyzing hundreds of thousands of specific positions across a person's genome. It is the same type of profile that consumer ancestry testing services generate when someone submits a sample. It is what powers ethnicity estimates and relative matching tools on those platforms. To search genetic genealogy databases, according to available methodology descriptions, investigators need a SNP profile generated from crime scene DNA. That profile would then be uploaded to the database. The system would find partial matches, people who share enough genetic material with the unknown profile to suggest a family relationship. From those partial matches, a skilled genetic genealogist would build family trees, trace lines of descent, and potentially identify the individual whose DNA was found at the scene. This is the framework. According to available record, it has reportedly been used to contribute to solving hundreds of cases, and it is reportedly what is being attempted in connection with this investigation. But according to publicly available expert statements, something is blocking it, and that something is specific. The person who went on the record to explain this publicly is, according to available records, not a peripheral figure in this field. She is, according to publicly available professional histories, the person who helped build it. According to her publicly available professional biography and multiple verified media appearances, she is the chief genetic genealogist at Parabon Nanolabs, the forensic genomics company that reportedly pioneered genetic genealogy for law enforcement. She has, according to publicly available records, personally contributed to solving more than 300 criminal cases using genetic genealogy. That reportedly includes homicide, sexual assaults, and cold cases that had gone untouched for decades. She has reportedly testified in court as an expert witness. She has reportedly trained law enforcement agencies across the country. When she speaks about the capabilities and limitations of this technology, according to available analysis, she speaks from direct operational experience, not theory, not academic speculation. She has reportedly sat at the keyboard, uploaded the profiles, built the family trees, and personally observed cases break open that had been closed for 30 or 40 years. She spoke on the record to TMZ about the specific technical obstacles reportedly facing this investigation. She named those obstacles precisely, she detailed the science, and she named the company reportedly working to solve it. According to available reporting, those were deliberate choices. Here is what she confirmed publicly, according to available reports. More than 50 million consumer DNA profiles reportedly exist in accessible databases. According to her on-record statement, she believes federal investigators would likely seek warrants to access the largest consumer DNA databases, potentially including those that have, according to publicly available company policies, barred voluntary law enforcement cooperation, despite the fact that, according to publicly available information, the three largest companies have reportedly barred law enforcement from using their databases voluntarily for genetic genealogy purposes. A warrant is, according to available legal analysis, a different matter. A federal court order reportedly cannot be overridden by private company policy. The legal process, according to available reporting, exists. The authority reportedly exists. The databases reportedly hold the profiles. And yet, according to available reporting, the databases have not been searched. Why? The answer, according to her publicly available statements, is technical, and it starts with what DNA evidence actually looks like at a real crime scene. According to available reporting, the assumption that most people made when they heard DNA evidence had been collected from inside Nancy's home was that the next step was straightforward. Run the DNA. Search the databases, get a name. That assumption, according to available expert analysis, appears to have been incorrect. Not because investigators are incompetent, not because the legal process failed, not because the databases are unavailable, but because, according to available expert statements, the DNA evidence may exist in a form that genetic genealogy databases reportedly cannot currently use. She confirmed this publicly. The DNA evidence reportedly recovered from inside Nancy Guthrie's home is what forensic scientists describe as a complex mixture. A DNA mixture occurs when biological material from more than one person is deposited in the same location, on the same surface, and collected as a single sample. When a forensic team swabs a surface in a residential environment where family members, visitors, contractors, and other individuals may have all left biological traces over time, the resulting sample can contain multiple people's DNA simultaneously. Their genetic material is physically mixed together in the same swab. Complex mixtures, according to available forensic science descriptions, are not unusual at crime scenes. Forensic laboratories reportedly deal with DNA mixtures regularly. Two-person mixtures, a victim and a suspect, for example, have established methods for analysis. When one contributor provided significantly more DNA than the other, scientists can often isolate the major contributor's profile. But when more than two people contribute to a mixture, or when the ratios between contributors are unclear, according to available analysis, the picture reportedly becomes dramatically more complicated. The genetic signals from each individual overlap and interfere with each other in ways that current deconvolution algorithms may struggle to separate. This is described, according to available forensic science literature, as a complex mixture. And complex mixtures are, according to available expert analysis, reportedly the point at which current forensic science, for the specific purpose of generating a SNP profile for genetic genealogy, runs into a significant obstacle. Here is why that matters specifically for genetic genealogy versus traditional CODIS searching. For standard CODIS searches using STR profiles, scientists have reportedly developed mixture analysis tools that work reasonably well even with complex samples. STR profiling examines a relatively small number of genetic locations, typically around 20 or fewer, which reportedly limits how badly the signals interfere. here. But SNP profiling examines according to available scientific description on hundreds of thousands of genetic positions simultaneously. These positions are spread across all 23 pairs of human chromosomes. When multiple people's DNA is present on each of those hundreds of thousands of positions may reflect a mixture of contributions from multiple individuals, building an algorithm that can, across all those positions simultaneously, reliably assign each genetic variant to the correct contributor on, and do so with enough accuracy that the resulting profiles are actually usable for genealogy database searching, is, according to available analysis, an unsolved computational problem in its current form. She confirmed this explicitly in her publicly available statements. This is reportedly not a resources problem, not a processing time problem. According to her public statements, it is an algorithm problem. The software that could reliably deconvolve a complex SNP mixture into individual contributor profiles does not currently exist in a validated, reliable, legally defensible form. Without a clean single contributor SNP profile, the equivalent of one person's complete genetic fingerprint isolated from the noise of a complex mixture, uploading evidence DNA to consumer databases reportedly produces nothing useful. The system is, according to available analysis, not designed to accommodate a contaminated multi-contributor sample. The matches it would return would be statistically meaningless, and so, according to available reporting, the pathway waits. But, and this is the part of her publicly available statement that changes the entire frame of the conversation, she did not stop there. She confirmed publicly that a company called Estra is reportedly actively developing software specifically engineered to solve the NSP mixture deconvolution problem. Not generally, specifically, fast-tracked, present tense. Active development, the word fast-track, according to available analysis, matters. It reportedly does not mean expedited on a standard research timeline. It suggests the development schedule has been actively compressed. The resources, the